So Philip Kai spends 25% of his time at Nottingham, but the rest, 75%, he works at the Toulouse Biotechnology Institute, where he has quite a large group. And these synthetic biology centres were formed just over 10 years ago now, so there were six UK synthetic biology centres covering all aspects, from health to plants, and our particular centre was focused on platform chemicals and industrial biotech, when in fact we are completely focused on single carbon gases, so mainly CO2 but also CO, and that's the feedstock we use, so we don't use sugars. So I'm kind of taking a step back, talking about butanol from sugar here, because it's kind of when I started my PhD 40 years ago. But I was doing this in the project with Vivek that we had here, so that's what I'm going to talk about. So what is butanol? Well, butanol is a full carbon alcohol. There are 4 isomers, only 2 of which are biologically important. That's N-butanol and isobutanol. And it's used for all sorts of things, for numerous chemical and textile processes. So I've listed a few there. It's involved in paint thinners, in lacquers, in enamels, in hydraulic and brake fluids, synthesis of 2-butoxyethanol, acrylic paints and perfumes. So it's used in all sorts of things. And it was, up until the middle of the last century, it was made by a fermentation process which was reliant on a bacterium, a Clostridium, an anaerobe, called the acetone butanol ethanol, or ABE fermentation process. And this was developed during World War I to overcome shortages of acetone which were needed for munitions manufacture. And a chemist at Manchester isolated this organism, Clostridium acetobutylicum, and developed this process. And then it was exported around the free world to generate the acetone that was needed for munitions. And it was in fact really the forerunner of the modern biotech industry, because it was the first fermentation process that was based on a pure culture, a pure sterile culture. And it was second only in scale to ethanol production at the time. So it was a very big process. And it can rely on 4 different Clostridia. So people always think of Clostridium acetobutylicum, but in fact there are spread throughout this genera of Clostridia here. Oh God. There are many Clostridia, 4 of which are solventogenic. They make solvents from sugar. They're saccharolytic. So it's actually a biphasic fermentation process. So in the first phase, the organism makes acids and excretes those out into the medium, acetic acid and butyric acid, and then at a certain point it switches over to make solvents. And then it reabsorbs those acids and turns them into butanol, acetone and ethanol in a ratio of 9 to 6 to 1. I'm going to list that again. If I could just have a look, I'll be able to see the button. Okay. So that is, okay, so that's shown here in the right-hand panel of the organism first makes acids, and then it switches over, reabsorbs the acids so the acid levels go down, and then exports the solvents into the media. So with the emergence of the petrochemical industry, obviously they started making butanol, mainly, that was the real aim, from petroleum, and that caused the ABE fermentation to decline, and so the last plants were closed down in the UK and the US in 1960. And the process carried on for a while in South Africa because of oil sanctions, but they eventually closed down in 1982, the last plant closed down. And then interest in ABE diminished through the 90s and the noughties. So, as I say, acetone butanol is now made from petroleum, and these just shows the processes by which they're made. And there are obviously lots of compelling reasons why one might want to return to the biological process. Clearly, there's the usual ones of energy security, the finite nature of fossil fuels, the environmental damage it causes extracting and using and exploiting fossil fuels. But of course, nowadays, there's this big push now on reducing greenhouse gas emissions, and obviously you have major greenhouse gas emissions when you make these chemicals by this route. And there's also a big push from society and from consumers to try and come up with green products. So I guess I got involved in the UK's Bioenergy Centre back in the noughties, and at that time DuPont and BP announced they were going to go into biobutanol production. And that was, to me, was the big turning point when it suddenly became sexy to start working on these bacteria. And we used to call them solvents, but then we started calling them biofuels. And whenever butanol is mentioned, people always say it's a better biofuel than ethanol. And there are obviously reasons why that is the case. It's got higher energy content than ethanol. Its low vapor pressure and its tolerance to water means that you can reduce caustic degradation of pipelines, so you can transport this through the same pipelines you do as petroleum. You can blend it with petrol at higher concentrations. It offers better fuel economy than petrol ethanol blends, and it can be upgraded to aviation fuel. So people always zero in on that when they talk about butanol. It's a biofuel, but really it's got tremendous use as a chemical. And when it became sexy to start trying to think about making butanol again biologically, people thought, oh, Clostridia, that's really hard to work with, so let's do it in E. coli or yeast. So that's what a number of companies did. So Gevo, one of the big examples, were founded in 2005 in the US. And then I've mentioned DuPont and BP announced this partnership in 2006, but that eventually turned into a company called Butamax in July 2009. And over the years, these two companies have progressed to generating different demo plants, and so they produce isobutanol because they're making isobutanol because they're doing this in E. coli. And then the two companies got locked into patent litigation. They fought for a number of years about who owned the rights to make isobutanol from E. coli. And eventually, Gevo acquired all of the Butamax patent estate in October 2021, that's quite recently. And Butamax are no more. Gevo are still going, but they haven't yet made any money out of making isobutanol, a familiar refrain. So most other companies in the world kept on with Clostridia. So most efforts were focused on trying to bring back ABE using Clostridia. So the earliest people were a company called Butafuel. So this guy, David Ramey, he drove a car across the US entirely fueled by butanol to prove that it was a good biofuel. Didn't really do a lot. 2003, Green Biologics were formed in the UK by an ex-PhD student of mine. 2005, a company in the US called Cobalt, same year that Gevo were formed. And then in 2009, Butamax went by the wayside. Green Biologics merged with Butafuel. A company in Scotland was formed, Celtic Renewables, say a little bit about them in a minute. And then Cobalt Biofuels transmuted into Cobalt Technologies, and then 3 years later they went under. 2019, Green Biologics went under. Butamax went under in 2021 finally, and then in 2023 there is this company in Scotland called Celtic Renewables. And they all got to various stages. They all tried to retrofit ethanol plants for butanol, but it all failed, and really they always seem to fail because of the cost of the feedstock. It was always the cost of the feedstock that brought them down. And that's just saying the cost of the feedstock in order to make these products, that the productivity they could get with those strains meant that it wasn't economic. And you can see that pie chart there is one from Green Biologics in the middle of the 2000s, showing the cost of making butanol from cornstarch. And you can see that 60% of the cost is the feedstock, but that's usually the case for fermentation processes anyway. So Celtic Renewables have based their system on the byproducts and wastes, and some of these really are wastes, of the Scottish distillery industry, pot ale and draft. And I'll say a bit more about them in a minute. But But strain engineering could address these issues of productivity, because if you could increase the productivity or maybe make more valuable products, then you should be able to develop a process based on biomass. So Philippe's lab and my lab over the years have published lots and lots of papers of technologies and analysis of Clostridium, how we might be able to move to revive the ABE process. And so here's a schematic of the genome of Clostridium acetobutylicum. And on here you'll see that there's a large plasmid called PSOL1, which is down at the bottom there. It's a megaplasmid, it's 192 kb, and all of the genes, the really important solvent-endogenic genes, are on that plasmid. So the ones that give ethanol, acetone, and butanol, they're all on that plasmid, PSOL1. Whereas the genes responsible for the acids in green are on the chromosome. So this is a result of a lot of Philippe's work with acetobutylicum. So he made a strain where he's made all of these mutations and made all sorts of substitutions to it to try and improve the amount of butanol made. And so the strain at the top with that big long list of genes that have been knocked out, so he's knocked out a gene involved in restriction modification, a gene, UPP, that he needs to knock out to use his negative selection marker. He's then knocked out the BUC and PTB genes, they make butyrate, so it doesn't make any butyrate. He's knocked out the CTFAB genes, so it doesn't make any acetone. He's knocked out the LDH gene, so it doesn't make any lactate. He's knocked out the delta rex A gene, so that's a regulatory gene involved in controlling butanol production. And then he's replaced a thiolase, which is a gene that takes acetyl-CoA to acetyl-acetyl-CoA, from, he's replaced it with one from E. coli, which has better kinetics to the Clostridial one, and then he's changed the HBD gene from, I think it came from Clostridium clervii. So having made all of those gene changes, he's come up with an organism that has got the most, the highest levels of butanol known from a Clostridium, and he's developed a process based on continuous fermentation. So it's making 550 grams per litre titer. That's making 0.3 grams per gram of glucose, and its productivity is 14 grams per litre per hour. And this performance level is equivalent, he believes, to that of a corn wet milling ethanol plant that uses yeast, and it holds the potential to be scaled up at pilot and industrial levels for commercial production of butanol. So what other process improvements could you bring about through engineering of the strains, which is kind of the sort of stuff we work on? So unlike rival petroleum processes, the ABE fermentation is not suited to continuous process technologies because it has this propensity to stop producing solvents, and it does that because it loses that big plasmid, that PSOL1. It gets lost by the culture. It's an autonomous element that can happen, and it no longer produces solvents, and that's called degeneration. And that was always a problem in the process all those years ago. So that's one thing you could do in terms of strain improvement. And the other thing is you could maybe improve the value of the solvents being produced, because acetone is not that valuable compared to butanol. So maybe you could produce something else, and what you could do is make an isopropanol, N-butanol, ethanol mix, or an IBE mix. So we tackled this as part of the project with Vivek, which was funded by BBSRC, Innovate UK, and the DBT. And so the obvious way to stop this degeneration is to stop that plasmid from being lost, and one way you could do that is you could integrate it into the genome. Now, we've previously developed some technology which is we call allele coupled exchange, or ACE, and with this technology we can introduce DNA of any size or complexity into the genome. And it's made possible because we have a plasmid-borne allele which we then couple to a chromosomal allele, and we create a new allele, and that's selectable. So we have a selection for this DNA to go into the chromosome. And the sequence of events in this process is controlled using homology arms of different length. And this allows us to define the segments of DNA to be more precisely and stably inherited at the designated genome. So I guess I was just going to show this because it really is ever so powerful and it's so simple, and I guess a lot of people don't seem to quite get it. So I've got a setup here. I'm showing the chromosome at the top and I'm showing a plasmid, an ACE plasmid below. In the chromosome there's a PRE gene which I point out, and that enables the organism to make uracil, so it doesn't require uracil. So it's a uracil prototroph. The plasmid, on the other hand, has an allele of the PRE gene which lacks this 5' end of the gene. It's dysfunctional. So if the chromosome had that, it wouldn't be able to make uracil. And then the other factor you need to know is that if an organism has a functional PRE gene and you give it fluorotic acid, FOU, then it converts it to a toxic metabolite and dies. So anything that is positive for the PRE gene is sensitive to fluorotic acid, and it's uracil plus. And the other fact you need to know is the plasmid carries a CAP-P gene, which encodes resistance to cyanophenicol, so TM resistance. So what we're going to do is we've created two homology arms, a large one, the right-hand homology arm, and a smaller one, the left-hand homology arm. And because the right is larger, it always integrates with that one in preference to the smaller one. And so when you put this plasmid, this lower plasmid, into the cell, and it carries a defective replication function, and you select for cyanophenicol resistance, the CAP-P gene, then all the colonies you get, or the ones that grow fast, will be integrated at that long homology arm, always. And then you can then plate the cells onto fluorotic acid. And the only cell that can now survive is the one that becomes a mutant for PRE. And so that causes excision at that second homology arm. And so you'll see the homology arm from the plasmid will now flip across onto the chromosome, like that. And then this plasmid will lost because it doesn't replicate very well. It's soon gone. So you've now got your PRE gene into the chromosome. And you need to do this because we're going to use this gene now to integrate other DNA into the chromosome. So to integrate at that site, we do the opposite. We take a plasmid that now carries another allele, and you can see that this one has a functional 3' end, but it has a non-functional 5' end, so the one that's sat on the plasmid. And so then we can clone cargo into that site between the two homology arms. We then transform the plasmid in, it integrates at the large one, and then if we plate this cell onto media without uracil, it cannot grow because it's got a PRE mutation. But there's one set on the plasmid, so if you get excision, you'll see that that allele now swaps across onto the chromosome, and then this plasmid is lost. So you now put your cargo into your organism. And the whole point about this is that an organism that is a PRE mutant cannot restore that without this process happening. It cannot revert back to alpha. It can't. It's missing DNA. Where's it going to get it from? So it has to get it from somewhere. So this is so powerful. When you do this, every single colony you get that grows without uracil is what you're after. So the problem is that's a little tiny plasmid, and anyone who does molecular biology will know you cannot clone a 192 kb plasmid into a little tiny cloning vector. It's very, very difficult to do. So in essence, what we did is we reversed the process. We took our small plasmid and we inverted the homology arms so that when they integrated into the chromosome, you would into the megaplasmid, the p1, it would look as though you cloned it. That's what you'd get. So it's not so obvious from the diagrams that show this. This will be in the paper when it comes out soon. But essentially what this diagram is showing is that that plasmid at the top, I wish this would work. It's so indistinct. So this plasmid up here at the top is actually, that's the ACE plasmid, and it has two homology arms that flank the Rep-A gene. You can see there's something called Rep-A, that's the replication function of the PSOL1 plasmid. And so we're going to now integrate that ACE plasmid in such that that Rep gene is deleted and replaced by the homology arms for PRE, which are in blue, and the point about that is that that integrated plasmid can no longer replicate, so it will be lost. But we've given it these homology arms, these PRE homology arms, which enables it to integrate at that PRE gene that we've just created, and we can select that with uracil. So we can do two complicated double crossover changes in a single two-step selection. So first of all onto uracilmycin, because we're selecting for the rep protein to drive that resistance, and then we're selecting for uracil plus. So we do it in 2 stages, and so we end up with that, and that's what we wanted. We wanted that. We want the PSOL1 into the chromosome. So we did that, and we ended up with it. And so this is just showing what you see when you do a continuous fermentation process. And on the left is the wild type, and you can see that after 20 hours, it's lost PSOL1, it's gone. And so it's no longer producing solvents, the green solvent ends, and the acid, orange, goes up. Whereas the integrated form on the right, we get continuous solvent production. It doesn't drop off at all. So by integrating PSOL1 into the organism's chromosome, we've now stabilized the process. So this organism will not degenerate, which is what happens if you use the wild type in an ABE fermentation. And then the other thing we did was we actually then used ACE again to integrate the genes you need to make isopropanol. And doing that, we could make an isopropanol butanol ethanol mix, which can be used directly as a fuel and is more valuable than if it was acetone. And so this is just showing that if you compare the production of IBE to any other system that's been published, then this system is the most productive. You can see that it's 15.5 grams per litre per hour. So this is the most productive clostridial system that's been generated for making IBE. And so the successful reintroduction of ABE fermentation process will require the identification of low-cost carbon feedstocks, because that is what's caused the process to fail in the past. Something like bagasse would have great potential for this, but numerous companies have tried this. So Cobalt and Green Biologics, they all tried to do this, working with China and Brazil, and China themselves have tried to do this, and it's all ended in failure because the feedstock price is too much. But it might be using improved strains like I've just described, giving higher product levels, titers and productivity. That might be the solution. And there are 2 caveats to that. What's the best strain to use, and which GM method should you use? Now, people always assume acetobutyllicum is synonymous with the acetone butanol fermentation, but in fact, in some preliminary work we did with some Brazil some years ago, we showed that if you took energy cane hydrolysates, then acetobutyllicum wasn't very good in terms of the amount of butanol produced. In fact, the best strain was this organism called Saccharoperbutyl acetonicum. We always call that the PER strain because it's such a long name, but it is the best strain in our hands on energy cane hydrolysate. And in fact, these companies never admit what they're using, but Green Biologics were using that strain, and so was Cobalt. So they were both using that strain. Celtic Renewables, on the other hand, are using acetobutyllicum, and they do seem to admit that. And then the other question is what GM method should you use? Because you're going to have to use GM if you really want to make productive strains. And CRISPR-Cas is the most rapid and effective way of making mutants in most organisms. But a CRISPR license is expensive, the patent landscape is complicated, and companies want to avoid using it if they can. So we've developed a non-CRISPR-based method, which we believe is as good, if not better, in many ways. So we've recently founded a spin-out company, so if anybody's thinking of using CRISPR, they should contact us. And I should just finish by acknowledging the people who've worked on this over the years, and particularly for this latter butanol work, the Innovate DBT Nuke Waste project.